Hello and welcome to our very important discussion on endocrine regulation. Today we will be talking about the two different second messenger systems. So, we talked in the past about how, in order for signal transduction pathway to manifest, we need a messenger, we need a chemical signal that must bind a receptor like a lock and a key formation. And once that signal, that stimulus, that hormone binds that receptor, the receptor is going to undergo conformational change that will elicit the beginning of a cascade that is a signal transduction cascade that will eventually lead to a response in the target cell. So before we begin, we must define really well what is a target cell. The target cell is the destination of the hormone. The hormone is trying to go and elicit a target response in the target cell. And that hormone, that messenger, that first messenger, is going to bind the receptor on the target cell, and upon binding to the receptor of the target cell, a signal transduction cascade is going to begin that will ultimately end in a target response within that target cell. So we have two second messenger systems. So in order to understand the differences between the two second messenger system, remember, I've always told you, whenever you want to understand the differences between two things or multiple things, always begin with the similarities, because the similarities are the stepping stone to making you understand what are the differences. So let's begin with the similarities. What are the similarities between our two different second messenger systems? Well, here we have the calcium calmodulin second messenger system. So we have the calmodulin, calmodulin second messenger system. The second system that we have over here is the G protein coupled receptor system. The G protein coupled receptor system. The similarities are both of them utilize a second messenger inside the target cell. So neither of these two systems will have a messenger that's able to traverse the cell on its own and be able to localize from the outside of the cell, from the extracellular environment to the intercellular environment. So we will always have a primary signal. We will have a primary signal. This primary signal is our hormone. This is our stimulus. This is the stimulus that is trying to come and bind our receptor, our target receptor in our target cell in which we're trying to elicit a response. So this is our signal, is going to bind a receptor. The same thing is happening on the second messenger system too, the calcium calmodulin system. You have a stimulus, you have a hormone that will bind the extracellular receptor. Now, you must remember that we have two types of signals, two types of hormones. We either have a steroid hormone or a non-steroid hormone. So hormones are classified into two different categories based on the structure, based on their anatomy. So we either have a hormone that is a steroid hormone, and that one is very easy in the sense that because it's a steroid hormone, it is built in the form of a lipid, and the lipid bilayer, so that requires an understanding of the structure of the cell. The cell has an outside membrane that is a double layer leaflet, and it has a hydrophobic core. And that hydrophobic core will prevent any hydrophilic or polar substances from transversing it and getting through it. So the steroid hormone, because it's a lipid and it is nonpolar, it will easily cross. So that means the steroid hormones do not ever need a second messenger. They will do everything on its own, on their own. So the steroid messenger will cross. So the steroid hormones utilize a first messenger. The steroid hormones never utilize a second messenger system, because we only utilize a second messenger system if we need somebody to carry the signal because we are unable to cross the cell membrane. So that is utilized with polar substance, with non-steroids. So the non-steroid system, they utilize a second messenger because themselves they cannot cross. Remember, your home, you can open the door, you have the key, and you can enter into your bedroom and open the safe and take the money and walk out. But if you send me to your home, I cannot do that. So I can knock on the door, and then someone from your family, your spouse, or somebody from your family, I will tell them, and then they will be the second messenger who will enter into the bedroom and open the safe and bring the money, and then I will deliver it to you. So what's happening here is the same thing, where you have steroid hormones are able to cross the membrane. They can get through the door, and then they make their way not only through one door, but actually two doors. They can get through the lipid bilayer, the outer capsule, this outer cell membrane, and then they can swim through the cytoplasm. Then they cross the nuclear membrane, and there is another medium known as the nucleoplasm. So there are two mediums: the cytoplasm, and inside the nucleus, the nucleoplasm. So the steroid hormones, they can cross the cross the lipid membrane, swim through the cytoplasm, and then cross the nucleoplasm, and then in there they will bind a receptor, and the receptor with this steroid hormone, they will mobilize. This is known as the nuclear model, where inside the nucleus you will have this mobilized receptor, and it will bind to the DNA and transcribe for us an mRNA that will later be translated into the ribosome to give us the target proteins that will elicit the target response, being the synthesis of extra uh, protein channels that will allow the influx or increase the permeability to some particular type of ion, or maybe the synthesis of the synthesis of some enzymes that are able to perform and catalyze some very important biochemical reactions in the cell. That is steroid hormones. But here we're no longer dealing with steroid hormones. We're dealing with hormones that are non-steroid. Non-steroid meaning they are disabled and unable to cross the cell membrane. They cannot cross this yellow lipid bilayer. So what they do is they bind on the outside, then a second messenger will carry this a second messenger will carry the signal. So we have two second messenger systems we are responsible of. So let's begin with the first second messenger system, known as the G protein coupled receptor. What happens in the G coupled protein receptor system is that you have a first messenger, and this first messenger hormone will bind like a lock and a key, in a lock and a key formation, to this G protein coupled receptor. Then the G protein coupled receptor will undergo a conformational change. So step number one is the binding of the hormone to the receptor on the target cell. Then the receptor on the target cell in the second step will undergo a conformational change, and that conformational change will lead to this G protein that is coupled to the receptor to bind GTP, guanine triphosphate. That is the third step. The third step is the binding of GTP to the G protein. The GTP binding to G protein will lead to the activation of a very important enzyme known as adenyl cyclase. When adenyl cyclase is activated, it's usually inactive, but it only becomes active when G protein is activated by the binding of GTP, and the G protein was activated by the binding of GTP due to the conformational change that took place in the structure of the G protein coupled receptor, and the G protein coupled receptor only changed its conformation after the hormone successfully binded like a lock and a key with receptor. Then the adenyl cyclase is going to be activated, and when adenyl cyclase is activated, that's its job. You see, I have two negative signs here, two minuses, two minuses. This shows you the effects of adenyl cyclase. What it does is it takes adenine triphosphate. So adenine has three phosphates, one, two, three. What adenyl cyclase does, it removes two phosphates and leaves one behind and creates for us cyclical adenine monophosphate. It used to be adenine triphosphate, now it's adenine monophosphate. This adenine monophosphate is my second messenger. This adenine, this cyclic AMP, this adenine monophosphate, is the second messenger that will relay what my first messenger is ultimately trying to do. And what it will do is it will go and it will activate an enzyme known as a protein kinase, and the protein kinase usually leads to phosphorylation. And then this adenine, this now activated, it used to previously be inactive, but it becomes activated when cyclic AMP is initiated and activated. Then the activated kinase protein will go and will phosphorylate. Phosphorylate. It will phosphorylate phosphorylate other enzymes, and these other enzymes, they will convert substrates into products, and these products can ultimately perform and catalyze some reactions that will ultimately propagate very important biochemical pathways. That's the first system, this adenyl cyclase system. The second different messenger system is that the calcium calmodulin. So here we're no longer dealing with a G protein coupled receptor. With the calcium calmodulin complex, we're no longer dealing with a G coupled protein receptor. We're no longer dealing with adenyl cyclase. We're dealing with calcium calmodulin binding. So we have inside the cytosol, we have an inactive calmodulin. You see, there's a cross, there's a X because it's empty. What happens is we begin with the same first step where we have a first messenger. You see, before the interaction happens, you have calcium deflecting, it cannot enter. This here represents a closed calcium channel. This closed calcium channel will deflect any calcium that is trying to enter. It will decrease the permeability. It will decrease the permeability of calcium. But what happens is when this hormone binds to its target receptor, what happens is we now no longer have a G protein coupled receptor. The binding of the hormone to the receptor will lead to the opening of these closed channels. So when these closed channels are now open due to the binding of a lock and a key formation with this hormone, with its receptor, now they're no longer going to deflect calcium. Calcium is going to smoothly enter down electrochemical gradient from the extracellular environment down to a diffusion gradient, down chemical gradient, down charge gradient, from the extracellular space to the intercellular space. And once calcium enters, now this inactive calmodulin is no longer going to be inactive. It's going to bind this calcium. So calcium will bind the inactive calmodulin, converting it to an active calcium calmodulin complex. And this calcium calmodulin complex is going to go and activate or inactivate. This is very important. Here with the G-coupled protein receptor, cyclic AMP is always going to activate, plus, 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 activate, activate. But calmodulin depends on the system. In one instance, it will activate an inactive enzyme, or it will inactivate an active enzyme. So when it activates an inactive enzyme, we will have substrates being converted into products that will lead to a target response. But if the calcium calmodulin complex leads to an inactivation of an enzyme, that will now decrease the conversion of substrates to products, and that will be the target response that we initially want. This is very important. And if you have last thing I want to leave you with is that in both, so the similarities, we talked about the differences. The similarities are in both second messenger systems, the nucleus was unbothered, it was not touched. You will never see, like in the steroid hormone, that you will have inside the nucleus, you will have the nucleoplasm. So the steroid hormone will swim through the cytoplasm, then it will the steroid hormone will first cross the cell membrane, then swim through the cytoplasm, then cross the nuclear membrane, then swim through the nucleoplasm, then bind the DNA inside the nucleus and transcribe for us mRNA. Then the mRNA is going to be uh, translated into proteins, and the proteins will give us like like more ion channels to increase the permeability of some ion, or maybe give us some proteins, and these proteins are going to become enzymes that will catalyze some reactions. But in the second messenger systems, the nucleus is unbothered, it's untouched. The nucleus is not involved. So that is universal with all second messenger systems. However, there's an exception. There is an exception to that rule, one exception. And the exception is the thyroid hormone. The thyroid hormone is an amine hormone that is built based on the structure of tyrosine. That thyroid hormone, although it is not a steroid hormone, it is a non-steroid hormone, but what it does is it has the ability to cross into the nucleus and bind an already bound receptor to the DNA. So with the steroid hormone, you have the receptor separated from the DNA, waiting for the steroid hormone to come, and then they will mobilize. That's why it's called the mobile nuclear receptor model. But with the thyroid hormone, the receptor is not mobile. The receptor is already bound to the DNA, and it's waiting for the thyroid hormone to come in and to transcribe for us mRNA complex, and then the mRNA will later on be translated. So the exception to the rule is that only the thyroid hormone is able to cross into the nucleus. But all the other second messenger systems, all the other non-steroid hormones will have nothing to do with the nucleus. It is only the steroid hormones that will be involved in the nucleus. Thank you very much.